chap. And eventually, we attract him down here for a PhD. And these are the production of Nicola in 2 years' time. And using some specific plan that is used in the population here, looking for the effect at the molecular level, looking for the interaction with some specific drug used in HIV, and the effect also in terms of pharmacogenomics. Once again, this is a typical example here. Do not necessarily use the same approach like anywhere else. Ask also from time to time your own question relevant to your population. So the last section of the talk is not research, but this is real implementation. A couple of years back, when we started to work in Cameroon originally, where we are genetically from, we realized that there were a few patients for which we were not able to provide solution. These patients have a condition called disorder of sex development. Because of our connection with Switzerland and with France, we bring down there a few friends with us. From Geneva initially, Pierre Lepoultre here. She was the head of pediatric surgery in Geneva and also the former dean. From Lyon, France here, Pierre-Yves here is a pediatric surgeon working in France. And we try to establish a program to provide at least, first of all, to understand what is the panel of the mutation. Second, provide some medical and some surgical option. What have evolved from that program is that for a condition called congenital adrenal hyperplasia, we know exactly what are the variant of the mutation for our population, the location, where it's frequent for what gene, and so on. Most importantly, we needed to have those specific mutations to provide surgical treatment and medical treatment for some patients. So this is a little girl that was born, and nurse sister cannot say if it is really a girl or really a boy in certain way. So we were able to provide a precise diagnostic and also to provide a restoration of the anatomy of this little girl that will grow like a girl and have kids like any other girl. And for some cases, more complex cases, this young man have breast development and he don't know what was happening, but he also have a penis. So with the program, we were able to show that this guy have a very complex condition called ovotesticular GSD. In the blood, he have to excess like a girl. He cannot produce a specific gene that allow a formal, not at least in the blood, in the peripheral blood, we couldn't detect a sex determination of chromosome Y. And on endoscopy, we could show that on one section, one part of his body, he had an ovary, on the other part, he had a testis. And after surgery, removing the ovary and also giving some androgen, we were able also to remove the breast because he was self-identified as a boy. He was known as a boy. Every single morning, he would wrap his chest so that the breast will not appear, so to hide it. And he'd been living like that for 14 years. So this was a very, very emotional case because the mom was crying, the guy was crying during the consultation. When we sorted it out, the next time when we saw him for the control, at this time he just passed his ordinary degree and he was smiling as ever. And once again, I'm just trying to showcase here, genetics is not just for rare conditions, it's not just for fancy science. Your real genetics for many people are suffering in silence because of some genetic condition that cannot be handled in our environment. So maybe also to say that this project alone have shown that genetic, the beautiful and the beast is the way we say it in English. I know how to say it in French. The beast and the beauty, right? Yes. Oh, the beast and the beauty and the beast. Okay. So it's the proof of concept that beauty and the beast can live together. Surgery and genetic can work hand in hand for the good of people. Beauty and genetic, of course. And this is the world map. And some of you might see something relevant to it. Sister Dorothy, you have to be kind to me, right? Sit down. So this is a world map showing innovation and world related to genetic research. And once again, you see that Africa is not present on this map. There is some part of Africa that are present. This is South Africa because of the work of people like me, probably. And but you can see the bottoms of Africa disappear completely. So my challenge in this program is that please, in 10 years' time, let's try to draw this map again. And this is a call for all the students in, be aggressive, be productive, be enthusiastic, so that we change the wall of the map and have exactly the place we should have on this map in a couple of years. We need to address three main things. We need to train and train again. We need to convince our politicians that genetics is important. We need to have technologies that is relevant to our community. If the electricity is not working, let's just have a backup behind and so on. When we started the first genetic diagnostic in Cameroon, I did it in my kitchen. I don't wait for me to have a lab. So I was moving a trolley, put my centrifuge, extract my DNA, go back, move the PSA machine, do it. We can do it with determination. We can do it if we have a strong commitment to do it. Of course, the data present here have been done by a lot of people behind here, all young, all students, and these are the list of students that have been involved in the project up to now. Valentina is the first PhD student that graduated from my group, and he's now postdoc in the WRC programs. I'm very, very happy of the work and the achievement he's done so far. And I hope that the other students working either on hearing loss or eventually on sickle cell disease will follow the same path. And I hope to have convinced you, I hope to have expect from you that genetics alone will not save Africa, but without genetics, some of our problems will never be solved. Thank you very much.
Thank you, Ambrose, for a very nice and challenging talk. I think I like the statement saying that we need to find our own mutations. So I'm sure the young ones have been challenged. We have a few minutes for a couple of questions.
Ambrose, once again, really beautiful talk. So in terms of the genetic diagnosis, I just wanted you to touch, if you could, on the collaborations that obstetricians bring to the table, because obviously,
yes,
if you're doing prenatal diagnosis, I'm assuming whether it's a coronary sampling or whether it's amniotic cells, the importance to have a collaborative effort that
yes,
involves OBGYNs, and one thing you didn't touch on was pre-implantation diagnosis. If you're going to do that, also you need our colleagues who actually interact with the mothers. Could you just touch on that?
Yes, I think that's absolutely. I think you probably work in a prenatal diagnostic setting, because I can hear from what you said. Initially, when we started this practice, when we arrived in Cameroon, we had no idea what was the map of expertise locally. We didn't even know someone could do it. But for a random event happened, we met the obstetrician. In fact, that was trained in fetal medicine in France, but that never had the opportunity to practice. And the second random event happened that the first couple that we had a child that had sickle cell anemia. And both couples were statisticians, so they knew the maths very well. And we said, okay, this lady is well trained, but have not been practicing it for some time, so you're going to take the risk of an amniocentesis or a chorionic valve sample that may lead to a miscarriage. I have not yet practiced a lab here. You're going to take a risk to have a lot of problem when eventually we are doing the extraction and so on. What happened that they took the risk. We took the amniotic fluid. No, we start with chorionic valve sampling. When I had the sample in the lab, that was the kitchen, I realized that I didn't have an inverted microscope to screen, because normally when you have it, you need to screen to take the villi from the fetal site. Okay, I look with my eyes, say my eyes is probably good enough with a spectacle, and then I tried to screen the way I could, and I extract the DNA from it, and eventually I had a result. But can you really give that result back? Now I get back those two guys and say, no, I don't trust this result. We will need probably to take amniotic fluid. In mature amniotic fluid, the advantage is that you have the population of at least 5 cells. The cell from the mouth, from the urinary tract, from the gastrointestinal tract, from the respiratory tract. So the rate of wrong positive, the false positive is very low. If you analyze your amniotic viral sampling, if the baby is heterozygote, it might be the mom that you have analyzed. That's the thing. So eventually we perform again. I had exactly the same result, but I was still scared to give back the result. What I did now, I took the blood from the mom, extract the DNA, send the 3 samples to my colleague in Geneva, and asked them to do the parent origin of the sample, viral sampling, and also amniotic fluid, and it came back, confirmed the result. It's only that we are able to give back the result to the parent, but still say, I need to see this kid at birth to confirm. So when the child was born, I took the blood again, do electrophoresis, and do molecular diagnostic again to confirm that we were right. And progressively, we start the collaboration with these geneticists, with these gynecologists, and up to now, even now in South Africa, she will take the amniotic fluid in Cameroon, and I will analyze in my lab in South Africa, and the things have been working very well.
Okay, so I'm Kojo Asamoah from the Gucci Memorial Institute. This is really extraordinary work. It's very impressive. I just have a question on ethics. I'm wondering whether you've had any challenges in terms of ethics, or because looking at the genome manipulation and working in Africa, as we all know, I'm just wondering how ethics has been, your personal experience as well as that of the area in general.
I think the question is very wide, in fact, ethics of genetics in general and ethics of practicing genetics in general. I think I will probably go take one aspect, which is practical ethics, not necessarily medical ethics or research ethics. Regarding one of the examples that we've shown here, when we started to introduce it, of course, people started to be and when we started to work on it, we contacted the National Ethics Committee for the Ministry of Health and say, what is the law regarding genetic diagnostic before birth and termination of pregnancy? It happened that in our law, you can stop a pregnancy for medical reason if the life of the mom is challenged, not the fetus. The life of the mom is challenged. Is a fetus affected by sickle cell of anemia challenging the life of the mom? The answer to that is no. So there was no ethical ground to use that law for that. So, but in the same law, they also say if the health of the mom is challenged. So it wasn't possible to use that law to practice at all. With the head hematologist, that was a sickle cell patient, and that co-supervised the thesis of Valentina with me, was pro-introducing it, but the head of the national ethics, that was a hematologist, bizarrely, adult hematologist, was against introducing diagnostic before birth. But one event happened that helped the discussion. There was a mom that had a child after in vitro fertilization and had a child that was multiple malformed, and the child eventually died. And that mom attempted to commit suicide. And her words was that I'm not able to have a child naturally, but the only one that can have artificially is a monster. That was the typical example where the health of a fetus challenged not only the health of the mom, but the life of the mom. And that specific case helped in the sense that for family that already had child with sickle cell anemia, that was already living the heat of sickle cell anemia, and we had data to show that there was a huge burden in the family, the middle line was to at least offer the diagnostic for family that already have a child with sickle cell anemia. So, and that's a little bit the way we do it this way. But having said that, I'm very uncomfortable with that practice for personal reason. I do think that I never have an interview about that practice. I never accepted to have a television show on that practice, not at all, because I do believe that what we should do is to look for solutions for patients that are there, because they will always be there.
I think so many people want to ask questions, but because of time, Ambrose is our run. Shall we please give Ambrose another one?
Thank you.
So we are moving to the fellows. The next person to give a talk will be Daniel Kipo. Daniel will speak on validation of candidate mutations in Plasmodium for resistance to the anti-malaria drugs, piperacin and lumefantrine.
Good morning, everyone. So my name is Daniel Kiboy, and I'm one of the postdoc fellows in WASBIP. And I'm going to present part of my work that I've been doing and part of the work that I'll be doing in WASB. And my mentor is Professor Faith Osiel at Wellcome Trust, Cambridge. So I just want to introduce this first. And in Africa, it's real now that we are using ACTs, or artemisinin-based combination therapy. And there are quite a number of them. I've just highlighted 3 of them. And the key thing is here to highlight is we are also working in high transmission areas. So there are 2 things. One, we have artemisinin-based combination therapy, where we have 2 drugs, and they have a mismatched pharmacokinetics. One is actually eliminated from the body very fast. The other one is actually left trailing. And bearing in mind that we are in high transmission areas. So we are actually receiving constant or frequent infectious bites. So the second infection, the third infection, actually meets a subtherapeutic dosage. That actually is the one that actually produces the pressure for selection of resistance. So when we focus on this, we started this work some times back. And we wanted to run ahead of the parasite. And the key thing was the pressure, at least in the high transmission areas, will actually primarily be subjected by the long-acting drugs. In this case, you'll be calling like lumefantrine, mefloquine, or piperaquine. So we ran ahead of the parasite, and just to highlight a number of things is, I used a rodent malaria parasite to select drug-resistant parasite. And I selected drug-resistant parasite by subjecting the parasite, by infecting mice first, and then subjecting the parasite to continue to increasing drug pressure based on how the parasite responded to the drug. And to highlight is this first passage means just mechanical passage where you infect mice, treat with a particular dosage, here with 6 milligrams per kg. And then depending on how the parasite grew, we proceeded on increasing the dosages. So initially the parasite was actually not responding so fast to the drug. But this actually shows the number of mechanical passages. So within 48 passages, we had a parasite that was highly resistant, or call it modern resistance based on the classification, that you could actually not kill even if you applied 52 times fold the initial dosage that we had. So this means that we had a resistant parasite, but this is uncloned parasite. But the most interesting bit about this was we wanted to test how this resistant parasite, that is lumefantrine resistant parasite, how it respond to mechanically and also chemically related and also unrelated drugs. And we tested against piperaquine, dihydroartemisinin, and also primaquine. There are a couple of others that I have not included. For the sake of this presentation, I've just highlighted 3. So, and you actually see that this parasite, actually lumefantrine resistant parasite, was sensitive to piperaquine. But there was cross-resistance between lumefantrine resistant parasite with DHA and primaquine. So to highlight on the next selection of piperaquine resistant lines, so within 27 passages, we had a 63 folds resistant parasite, meaning that you could, even if you apply 63 times resistance, you could not kill that parasite. And I want to highlight also, this was uncloned at this point. So now, we also wanted to test the cross-resistance profile. So this is what it shows. If you test with the piperaquine resistant line, and you use lumefantrine, the parasite was highly resistant to lumefantrine. So this means that if you have piperaquine resistance, you actually also lose lumefantrine. So there was also slight cross-resistance with DHA and primaquine. So, and this led us to first conclude that we have a multi-drug resistant parasite. So, but this is phenotypic data. So the key thing we wanted to establish is, what is the genetic makeup of this parasite? So, and before doing that, when you are actually selecting the resistant parasite, you are subjecting those parasites to, or you have a population of parasite, and different parasites mutate at different rates. But there is a common denominator that you want to select that could be a determinant of this resistance. So we first cloned we cloned this parasite by limited dilution. So you actually inject at least 0 or 1 parasite per mouse. And I selected three different passages. This is 60, 27, and also one of the AMP line, which is passage through mosquitoes. And then so in absence of the drug, so the parasite grew normally. But when you apply the drug after cloning, so you eliminated all the sensitive parasites, and this one in different mice. So you eliminate all this, the sensitive parasite. This means that we actually conclude that we have cloned this resistant parasite. So now we have a genetically homogeneous parasite that we wanted to take further. So we proceeded further, and with the help of Dr. Oliver Bilka and Julian Reina, we sequenced the whole genome of these parasites. And the key thing was to identify the novel SNPs and possible copy numbers that could be associated with piperaquine resistance and lumefantrine resistance. So we narrowed down on a number of mutations. And the reason is we actually looked at either proteins or enzymes that could be associated with either metabolism or transport of the drug, or other conserved proteins that could be of interest that have interesting mutations that we thought would be candidates for resistance. So we narrowed down on a number of them. For lumefantrine, we narrowed down on these 3, and then piperaquine, we narrowed down on these 3. I'll not dwell so much on this, but the one that highlighted in red is I'll highlight some preliminary data on this, on the actual deferodoxin NAD reductase. So, as Oliver mentioned yesterday, there are a number of resources that you can actually use to try to relate the phenotype, the genotype of, or the gene with the phenotype that you see. And actually some of them are plasmogene vectors. So we set objectives, and the key thing about this was to use 3 ways. One is to use the resistant line and try to repair the mutation in the resistant line. So the key thing is we want to revert the resistant parasite into sensitivity. The other way is to use a knockout mutant based on the mutation, or to use a mutagenized parasite or vector and introduce the mutation in the sensitive background, and then measure the drug profile. So, and the last one, of course, that possibly would use is the ones that are showing promise is also to use the CRISPR-Cas method and to introduce a specific mutation in P-falciparum and also measure the effect on the drugs. So I'll highlight one of them, which is the NAD reductive, the ferrodoxin NAD reductase of FNR that have. So, and it has been associated with also primaquine action, courtesy of Professor Steve and Giancarlo. And the key thing, what I did was first is to capture schizons and also prepare the vector. You transfect, and also once you inject in mice or infect mice, you select using either pyramidine or WR. And once you have the phenotype, then you can genotype with a panel of primers. The quality control primers, you test whether your vector is in the parasite, and also check whether you have integrated the vector into the right position. Also test whether you have repaired the mutation. Once you test whether you have repaired the mutation, then you do the drug profile. So to show this is the first one, the one I'm calling E10 plus R is the one that has been complemented, or the one that has been repaired. So the one I'm calling W is the wild type that is resistant. So we also needed to control for the effect of this vector. So we transfected the same vector in the sensitive background, just to control for the effect of the vector. So as you can see, there was no vector, of course, as expected in the resistant that has not been transfected, same as this. And so this is a detection of the vector, and this is a quality control primer for, especially for the wild type. So you have wild type. This is the gene that has been actually disrupted. So we also wanted to test, have we integrated the vector into the right position? So this is one of the primers, a couple of primers, set of primers that we tested. And this actually shows that we integrated the vector into the right position. So one of the primers actually needs to then adjust a gene from the position of our gene of interest. So the thing is, once we verify this, of course I sequence this and verify that I have repaired the mutation. But the key thing is, does it have any effect on the drug response? So this just shows a number of things. So I just highlighted a number of things. So the blue one actually is 10 milligrams per kg, and you can see the one that has been repaired, there is a shift in drug response. So this is a drug profile, but what is the quantity of reversing that resistance? So if you can see, against lumefantrine, the initial one, when I cloned the parasite, of course I would expect the resistance level to go down, but it's still within the threshold of being resistant. So the effect of repairing, which is this E10 plus R, we had a 27 point of ED90, or we call it IC90, the dosage that kills 90% of the parasite. So the effect of actually reversing or repairing the mutation was 70.9%. Against piperaquine, which I've showed, which is actually cross resistance, we had a 59%. So this is interesting. So I would want also to take this further to CRISPR and also focus on the other gene. I've just highlighted one, which is actually a premium, but this is also important to take this further, and some group in Liverpool are also doing this. We agreed to share some data. So they actually introduced a specific mutation in P. falciparum. So this is one of the key things. So sorry. Just to highlight on this and just to complete this whole thing is, having done all this is on drugs, and again it's on WASBIP. I would also want to focus on a number of genes that could be also involved in immunity. And this I'll be looking at transcriptome levels of asymptomatic individuals, that patients that are able to control malaria within their parastemia, but they don't get any clinical disease. So this is one of the things, and of course I would also want to do some editing of the genes, as the way I would also want to do is in drug resistance genes. So this is one of the things that I'll possibly be highlighting. So it's just one of the slides. And lastly, of course, is just to conclude and to acknowledge a number of people, and of course you also for listening, all those who have been involved in this study, my employer, Kemru Wellcome Trust, my mentors, and all the others. Thank you.
Thank you, Daniel. I think we have time for one or two questions or comments.
Daniel, when you infect, how many parasites do you put into the animal at the beginning? When you're starting to select by treating the animal, how many parasites do you actually put into the animal?
So once we do this, is, for instance, the initial thing that we start with is first is assuming your donor mouse is like 5%. So we dilute this to at least 1%. So the assumption is 0.2 ml of this 1% contains at least 1 times 10 into power 7 of the parasite red blood cells.
Right, about
1 times 10 into power 7.
10 to 7. Okay.
Yes.
So the reason I ask is that this kind of experiment is done very often, where you add parasites, and in many